Hey guys, welcome back to AP Bio. Today we're going to get into transcription and translation, which is what happens when DNA is converted into mRNA and then eventually a protein. But before we get started, I wanted to have everything open that you'll need to be successful for class, just because I want to make sure that you're organized and ready to go. So first things first, if you haven't already, go ahead and open up the Chapter 15 notes. And then you'll want to dual screen between this video and these notes. Your independent practice is going to be a brief activity on sickle cell, where you'll get to practice with some of the skills we're learning today. And then you have your exit ticket. I'm also going to post a link to a video that I mentioned in our PowerPoint, but you don't have to go watch this video. That's optional if you're interested. So without further ado, let's go ahead and get started with our notes. So first things first, we have a catalyst. I'm not checking these, but I do want you to use this as an opportunity to get your brain ready for learning. So go ahead and pause the video and then try to answer these two questions, and then come back once you've done that. All right. So if you try to answer number one, what happens when bases and nucleotides get mixed up or switched? If you're answering just from your knowledge and you took general bio, you know that this is what causes mutations. Mutations cause different genetic disorders, or mutations can be advantageous and actually help an organism by making them run faster, see better, etc. Hopefully you were able to think of some genetic disorders that people are born with, and then maybe some that they encounter through their environment as well. So for our agenda today, we just did our catalyst. You guys will have some notes to take while we go through our slides. You have a quick case study. It should take you no more than 10 minutes, and then your exit ticket for today. So after today, you guys should be able to identify the steps in transcription and translation. These are the final parts of the Central Dogma, or how proteins are produced. You should be able to use a codon chart or a wheel to figure out what happens to an amino acid sequence. And then you should be able to explain how a change in one codon can result in drastic changes to an organism. And this actually answers that final learning target that you have for today. So a small change in DNA, specifically if we're talking about mutations in the LMNA gene, which causes Hutchinson-Gilford progeria syndrome, this is a change in one nucleotide. That's one base on a codon. And because of that one change, what happens is the mutation causes the production of the lamin A protein to be abnormal. This new protein has an unstable nuclear envelope, which we learned about in our second unit when we talked about cell biology. So this is what a healthy nuclear envelope would look like on a cell. This is what someone with Hutchinson-Gilford progeria's nuclei might look like. So you can see the envelope is twisted, it's distorted, and remember, structure affects function. So when the nuclear envelope is unstable, cells are more likely to die prematurely, which causes early senescence or aging. So this is just a very fancy word for aging. This is what someone with HGPS looks like in general. If you'd like to watch a video, there's a really great TED Talk by someone who's living with this disorder about how they can live a happy life despite the challenges that the disease causes them. So I'll be linking that video for you to watch if you want. That is not required watching. You can pause and watch it now. You can watch it after if you're bored. So now let's get into the notes. First thing you need to know is that your genes specify specific proteins or enzymes that get produced. The name for this is the one gene, one protein hypothesis. So what it states is that inside your DNA, the different genes found on the DNA are recipes for specific proteins. Those proteins are the building blocks of enzymes. And remember, enzymes are the catalysts. They're the ones that carry out drug metabolism and can speed up or slow down reactions in the body. Three important things for you to keep in mind when we're talking about these processes of transcription and translation is that our chromosomes are made out of our DNA. And segments of DNA, so specific strands or parts of strands that are, between 100 and 1,000 or more base pairs long, will determine the proteins that get produced. And it's the order and sequence of those that determines the protein made. So the protein decides our traits, which we learned about in our last unit when we talked about genetics. So your eye color, the different enzymes you produce, your hormones, even the hemoglobin in your blood, which helps to oxygenate cells, is all made based on the directions located in your DNA. And today we're going to talk about how that information goes from stored form in the nucleus in the DNA to a usable form in the form of a protein. The name for this idea that genetic information goes from DNA to RNA and then eventually a protein, is the central dogma. So a dogma is an idea that's essential to a specific field. And our central dogma in biology is that information goes from DNA to RNA to protein in order to go from storage to usage. This is an image of what that looks like. So you have a double helix, which we talked about when we talked about DNA structure. The double helix is too big to escape the nucleus, so it has to be converted into mRNA through a process called transcription. And then finally, it's translated on the ribosome through a process called translation into a usable form in the form of a polypeptide or protein. So in order to understand this process, we also have to understand RNA. There are only a few differences between RNA and DNA, which we talked about last class. So the first major difference is that RNA has a ribose sugar. So that's why it's called RNA, ribonucleic acid. Remember, DNA has a deoxyribose sugar, which is why it's called DNA. The second major thing is that there is no T in RNA language. Instead, the base uracil gets used. So any place where we would have had thymine, we now replace it with uracil. And then the last thing is that RNA is single-stranded. So it is not a double helix, it's one strand, which is why it's able to escape the nucleus. It's small enough to carry that message out of the nuclear pore where the DNA could not. So we're going to watch a quick video because this does a really excellent job of showing you guys what this process looks like. I suggest that you watch it once or twice, whatever you're comfortable with, and then we'll get into the details. So I'm going to play it in here, but I'm going to hide myself because you guys don't need to see me while this is going on. And then if you have any questions, of course, you can pop into office hours or just watch it again. I think it does a really excellent job of explaining the process and the players involved in it.
What you are about to see is DNA's most extraordinary secret. The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body.
It's a bit like taking the information off a hard disk on a computer and put it into memory, making it a real program that's running. So that process of DNA to RNA is like running a program, double clicking on an icon.
When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin.
Again, if that was confusing for you, feel free to go back and watch that again in this embedded video, but I'm also going to upload the link in case you need it, just so you can watch it again at your leisure. But let's go ahead and get into it. And remember, as we go through these notes, you guys can slow down the speed on a YouTube video, you can pause, you can speed me up if you think I'm going too slow, whatever works best for you and your learning. So first thing we need to talk about is transcription. This is when the mRNA is made from the DNA, and it follows the same base pair rules that we learned about with DNA, except in this case, uracil replaces thymine. So A pairs with U, T pairs with A, and C still pairs with G, as we learned about before. So you can see in this image here, the purple strand is the RNA that's getting produced. A pairs with T, C pairs with G, G pairs with C, and U will pair with A. Now, the molecule that's used to start this process is called RNA polymerase, and you can see it right here in this image. So RNA polymerase begins the process of producing RNA, just like DNA polymerase produced DNA in our last lesson. The next step in the process is translation. This is when we take the mRNA, remember that's the usable code that can escape the nucleus, and we build a protein from it. And the main players in this are the mRNA, which has the code, and the tRNA. tRNA is another type of RNA that's used to build the amino acid chain. The way it does this is it reads the code in groups of codons. So 3 bases here produce 1 codon. And on the tRNA, which is this molecule here, you have an anticodon, which is just the opposite. So we have CUU. Remember, C pairs with G, U pairs with A, so the anticodon would be GAA. Now that anticodon matches the amino acid at the top here. So leucine is the amino acid that would get attached to this protein strand based on this anticodon. Translation happens on the ribosome. Remember, we learned about the ribosome when we learned about cell structure. So the mRNA, that's this purple part here, is threaded through the small and large subunit on the ribosome here. As the mRNA goes through, different tRNA molecules come along and see if the codon on the mRNA matches the anticodon on themselves, on the tRNA. If the codon and anticodon match, then the tRNA will drop off an amino acid. As the amino acids get dropped off, they string together. This is a peptide, polypeptide here, or a protein. So remember, polypeptide is a fancy word for protein, and the tRNA carries the amino acids which build that protein based on the instructions on the mRNA. So every tRNA delivers one amino acid which binds to the previous one and eventually creates a protein for us. Here's an image that shows you what all of this looks like on an up-close level. So every gene starts with one specific codon. Remember, codons are groups of 3 bases, and that codon is the start codon, AUG. AUG attaches to UAC and adds the amino acid methionine. This indicates the start of our protein. So without a start codon, the protein can't be built. After that, you'll have other codons, remember those are these groups of 3, that match to specific anticodons on the tRNA, which then attach different amino acids to the peptide chain. When we get to the end, it will stop with a stop codon. There are 3 of those, which you guys will learn about reading in just a second. And from start to stop, we have all of these different amino acids getting attached. It's the order and number of these that determine the protein that's produced. So now we're going to read a codon chart to figure out what amino acids get added, because remember, the tRNA is bringing a specific amino acid every time. So every 3 bases on the mRNA is a codon. Remember, mRNA is just a short form for messenger RNA, and that helps me remember what it does because it delivers the message from the nucleus to the ribosome. Every codon specifies 1 amino acid, and then the chain of amino acids that get produced is the protein, for example, hemoglobin. So this is a codon chart right here. You have one in your notes too, and we're going to practice reading it together so you guys can do your independent practice. So this looks similar to the one in your notes, except the one in your notes is not colored. Feel free to follow along in those if you'd like. So we want to know the amino acids produced by these 3 codons here, or the strand of mRNA. Remember, we look at them in groups of 3, so we're going to look at these first 3 first, then these 3, and then these. So I'll do the first one for you, then you can try these 2 on your own, and then come back and check your answers. So for the first one, we have ACG. To use this chart, it's pretty intuitive because it's labeled for you. So the first base is one of these rows, the second base is one of these columns, and then the third base is specific to the row and column that you identified. So we're going to start with the first 2 letters to hone in on a specific box. So we'll start with A. We're going to go to this row here, because that's our first base and our codon. Then we have a C. So we're going to be in this column here, which means we're looking for the overlap between the row and the column. And in this case, it's A and C, so we would go right here. Now our third base is a G here. So then we would follow that back to the left to our specific box. We get ACG, which gives us THR for our amino acid. Go ahead and pause the video here, and then we'll go over the answers of these last 2. All right, so for our second grouping here, we have triple A. So we're going to go to A here, then find A here. So we're looking in this overlap, right, because A and A. Then we want our third letter, which is also an A here, which produces lysine. And then for the final one, we have GAU. So we go to G, we go over to A, and then we go to U, which gives us ASP. So this is really easy to read, but it tells us which of the 20 amino acids gets produced from each of the different groupings of base pairs or codons. Next up, we have a codon wheel. These are even easier to use, and on the AP, you might see this or you might see the table, so make sure you're familiar with both. But I'll go ahead and do the first example again. You can pause and try the other two, and then come back and check. So for our first one, we have AAG. We're going to start in the center on the codon wheel and then just work our way out. So we have A, G, which gives us lysine. Then you guys go ahead and try these two, come back, and we'll go over it together. All right, so next up we have UCG. This codon wheel isn't great because it has the T's in here, but remember T gets replaced with U. So we'll go U to C to G to give us serine, and then G to A to U, which gives us aspartic acid here. So if you see this wheel on the actual AP, they would give you guys the U letters instead of the T's. All right, so this is going to be in your exit ticket. I want you to go ahead and pause this video and then see if you can label numbers 1 through 8. In just a second, come back and we'll go over it together. All right, so we'll just go in order. Some of these were probably easier for you than others, but this is just a model of that transcription to translation process. So this here we know is the nucleus, this outline, because we've got this molecule inside, which is number 1. So number 1 is our DNA. We can tell because it's a double helix. Number 2 is a single strand with these on it, which tells us that it's mRNA. And we know that because it's escaping the nucleus here. Number 3 is pointing to 3 of these bases on the mRNA. And remember, a group of 3 bases is called a codon. Number 4 is a molecule that's bringing these things here, and it has an anticodon on it. So we know number 4 is the tRNA. Number 5, I just told you what it is, right? It's our anticodon because it's going to match 3 of these bases on the mRNA. Number 6 is the thing that the tRNA drops off. Remember, that's our amino acid. Number 7 is the place where all this is going down. Remember, in the ribosome is where translation occurs. So number 7 is the ribosome. And then number 8 is a string of amino acids, which we know is a protein. So if you struggled with that, get a new sheet of paper out, go ahead and try it again, come back and check your answers because you will need to know that for your exit ticket. All right, and our last little bit of your notes before we move into the IP, when we're talking about DNA, there's some parts that code for proteins, and then there's some parts that don't. So the exons are the parts that we've been talking about this whole time. Those are the parts that code for specific proteins in the DNA. Now, between the different exons on a gene, you'll have something called an intron. These are our non-coding regions. So because of that, the RNA has to splice or be put together. So it removes the introns. Remember, that's the part that's not coding for a protein. And then it splices together just the exons here to produce something usable for the ribosome. So the molecules responsible for cutting out these introns are called spliceosomes because they are splicing the exons together. This is where your notes stop. You can, if you understood everything from class today, stop watching now. But if not, I'm going to walk you through how to do this first part right here on activity 2 in your independent practice. So in this one, it's giving us our DNA, and we have to convert it to RNA. Remember, when we're converting DNA to RNA, the same rules apply from last class, except in this case, A is going to pair with U. We've got A to U, T still goes with A, C pairs with G, G pairs with C. So the only difference in our rules here is that A pairs with U. So if we go up here and we're labeling our first RNA strand, we've got CAC. So we're going to put G, because that's what pairs with C. A pairs with U, and C pairs with G. So the RNA produced from this strand is GUG. Now, if you go back to your note sheet, you have a codon chart right here that you can use for this next part. So we've got GUG, and we want to figure out the amino acid produced. So if we go back to our notes, we've got G here and U here, which tells us we're in this box. And then if we follow the G back to the box we're in, we know that VAL gets produced. I'm going to do one more for you, and then you're on your own. So make sure you're watching if you're still confused. So for this next one, we've got GTG. We know that G pairs with C. We know that T pairs with A. And we know that G pairs with C. So we get CAC. I'm going to leave this here so that you guys have that to reference when you're doing your independent practice. So now that we have CAC, which is the RNA, we're going to go figure out the amino acid. We can't figure out the amino acid from the DNA. So we go C over to A and then to C, which gives us HIS. So I'm going to leave these first two filled in for you. You guys, for your independent practice, are going to complete the rest of this sheet, and then you'll do your exit ticket. Remember, you do need to know what that image had on it, so make sure you go back and practice that if you need to. As always, you can pop into office hours with any questions, and thanks again for doing. I'll see you guys on Thursday or at office hours if you need help.